This is TWIS. This Week in Science, episode number 878, recorded on Wednesday, June 1st, 2022. How to make a big old science omelet. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with axolotls, spider, and poop. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. On Memorial Day, we were supposed to have been remembering the fallen soldiers, those who were called to serve and sacrificed all. But instead, another massacre of children with an assault rifle has people remembering that the United States is the most violent, dangerous place on Earth. 14 mass shooting events over the Memorial Day weekend. Another one at a hospital in Oklahoma just today. And as the yearly deaths by gun in the United States continue to climb, 45,222 in 2020 alone, we are far exceeding our fallen soldiers in war, eclipsing the lives lost by our troops in the 8 years in Iraq, 20 years in Afghanistan, and the Korean War combined in a single year. Approaching every soldier's life lost in the 20-year Vietnam War every single year. And why? Because guns are poorly regulated in the United States. States that are well regulated have the fewest gun deaths, but gun deaths are higher in the U.S. than anywhere else in the world. We use guns to commit homicide and suicide per capita more often than anywhere else on the planet. Guns are the #1 cause of death amongst children in the United States. Memorial Day is a day to remember our fallen. We should remember who that is, even as we enjoy another episode of This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. Hopefully this week will be more full of science than it is of bloopers, but you know, it's always good for our TWIS blooper reel, which I know somebody is putting together out there. Ha 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 ha. Welcome, everyone. I hope you really are looking forward to a good show of science, because I have brought liver transplants, electric evolution, and exercise. We're going to exercise tonight on the show. Ready? What do you have for us, Justin?
I've got, just replacing the liver is fine. I've got a regenerative model that can replace everything.
Everything?
Everything. As well as a major extinction event in history. Also, an ancient city that was risen from the waters, and an ancient city that was overgrown and discovered by flying lasers.
Flying lasers? Ancient cities and flying lasers. This is the next adventures of Justin Jackson. Blair, what's in the animal corner?
Oh my gosh. I have baboon courtship, spider courtship, and some poop. What else is new? Just a normal week in the animal corner.
Courtship, poop.
Right.
Well, it is definitely stacking up to be a lot of good stories as we jump into the show. I do want to remind you that subscribing to TWIS It's fairly easy to do. You can find us on all of the podcast platforms that you can probably think of out there. Just look for This Week in Science. We are on YouTube, Facebook, and Twitch, where we broadcast live every Wednesday at 8 p.m. Pacific time. We are TWIS Science, TWISCINCE.
TWIS Science.
Twitch, Twitter, and Instagram. And our website is twis.org. It's time for the science. Let's talk about livers. Well, replacing them with transplants. And okay, we've talked about transplanting organs before. What is one of the big problems with
Rejection.
Yeah, rejection.
another big problem?
Donors.
Yeah. Supply chain.
They gotta find a living. I guess, but not really for long.
Yeah.
And then you have to decide who deserves the organ more because there's not enough for everyone.
Right. So one way that people have been trying to solve this problem is through synthetic livers, trying to grow livers in petri dishes, trying to grow them in the lab, make them de novo out of people's cells to get past the rejection issues. But this is still kind of futuristic science. A different attempt has been to make the livers that would be coming from potential donors last longer and also healthy them up a little bit so that they'll be good, healthy, basically brand new livers ready for transplantation, not on a rushed schedule, but can be planned and actually transplanted as needed when needed. Fewer livers going to waste. How would someone go about doing this? Well, out of the University of Zurich, a team called Liver for Life has created a perfusion machine. And this perfusion machine does everything that the human body does. It gives it oxygen to replace the lungs. It pumps to replace the blood. It has a dialysis unit to perform the function of the kidneys for filtration. It's got all these aspects of hormones and nutrients and all the things that a liver would need to be a nice, healthy liver. And so when somebody dies there is a liver available, it could potentially be put in this perfusion machine for a number of days and kept healthy. And what they have shown in their new press release and their demonstration is that they were able to store a liver for several days outside of the body in their Liver for Life perfusion machine, and then transplant it into a cancer patient who was on the waiting list for a liver transplant, but probably would not get a liver in time otherwise. So they kind of said, hey, you're probably not going to get a liver in time, but we've got this other option for you if you want it. And it's been successful.
I like how you put that. We've got this other option. So you mean my only option is that other option? Yeah, actually, it's your only option. So take it or die. Up to you.
Yeah, you could take it and it could work, or it could not, which is the same as not taking the option. So,
but it did work.
Yeah,
it did work, and the liver that they had put into the perfusion machine was actually not a really healthy liver to begin with, but it came out a much healthier liver and was well accepted by the organ recipient. And they just published in Nature Biotechnology their work, where it shows that by treating livers in the perfusion machine, it's possible to alleviate the lack of functioning human organs and save lives. They also go on to say the interdisciplinary approach of solving complex biomedical challenges embodied in this project is the future of medicine. This will allow us to use new findings even more quickly for treating patients. And so this project was actually launched in 2015, and it's a little rolling. It's like a little rolling liver trolley.
It's like a little aquarium, but instead of fish or lobsters, it's, livers.
Livers. And the livers can go on to live happy and healthy lives in their new recipients.
Oh.
But I love that this gets rid of, the big thing is the time bottleneck. because when someone dies, you have a donor, right? And everyone has to get there at the exact right time, collect the organs that will go for donation. There has to be a patient ready to receive the organs within hours. And very often things go wrong, livers, hearts, things go bad, and things get destroyed accidentally, or because there isn't enough time to get from one part of a city to another part of a city, or from one city to even another city. So this could potentially get past a lot of those bottlenecks and fix the problem. Liver for life. Yeah, I'm a fan. What do you have, Justin?
Oh, here we go. I'll start with this one. This is proteins extracted from fragments of a prehistoric eggshell found in Australia. Confirmed that the continent's earliest humans consumed the eggs of 2-meter-tall thunderbirds. These birds that just went extinct about 47,000-ish years ago. This is in the journal Proceedings of the National Academy of Sciences. The eggs came from Genyornis, a huge flightless thunderbird with tiny wings and massive legs that roamed prehistoric Australia in flocks. Would it still be flocks if they're flightless? Could it be herds? I think it would be herds if it's a
maybe herds. What would you say for ostrich, Blair?
That's a great question. I still think it's a flock.
It's a bird.
Of the feathers.
Yeah. We'll figure it out.
Interesting.
Yeah.
Fossil records show that the thunderbird stood over 2 meters tall, weighed between 220 and 240 kilograms, that's a lot. That's a big bird.
Big.
They laid melon-sized eggs that were about 1.5 kilograms each. And so the earliest date that we think Australia got populated by current modern humans, about 65,000 years ago, would have been maybe the furthest back currently. These burnt eggshells date to around 50 to 55,000 years, and that's not long before this giant bird went extinct. And it was about the time when humans had spread across most of the continent, and these eggshells are actually found in locations all across the continent. However, there is no sign of thunderbird butchery anywhere in the archaeological record. So they were too big to hunt, but the nest rating of the giant eggs was a productive behavior, at least for the humans. They could get away with that, although it wasn't sustainable, because obviously the giant birds went extinct. Researchers are pointing out that the Genyornis egg exploitation behavior of the first Australians mirrored very much early human ostrich egg raiding activity. And that's something that's in the archaeological record across Africa, dating back at least 100,000 years. So the peoples who eventually populated Australia were probably not that far historically removed from people who were doing ostrich hunting in Africa. But the difference is likely the reproduction rates. The ostriches and humans coexisted with this egg-stealing behavior throughout history, but the same level of exploitation with this thunderbird by the early Australians may have been too much for the reproductive strategy of those birds, and that's probably why they went extinct.
So it was just that the birds weren't reproducing quickly enough, so there weren't enough eggs to feed the hunger of the growing population of humans in Australia.
That's one of the possibilities. Yeah.
Do you think it's kind of? I mean, if it's people, I'm going to say, well, people, they're gluttons. They like the big eggs. They're going to go eat as much as they want, not worrying about that kind of thing. But I also wonder, is it population balance versus, or is it, I don't know, what they wanted to eat?
But look at that. How could you take the Thunderbird's egg? We're looking at an artist rendering that I think is very, very cute. 2
meter tall Thunderbirds.
Yeah, so
very
adorable.
Scary.
So adorable.
Not adorable, scary when you think about that.
Both can be true. But I'm surprised that they weren't able to protect their eggs. I mean, ostriches are very good at protecting their eggs.
Mm-hmm.
Perhaps
their
that's
big
why
eggs.
ostriches still are here and thunderbirds are
And,
not.
and it, that may have been a thing as well. There's also that weird habit that animals have of learning to be afraid. It's not weird, but they learn to be afraid of humans to an extent. Where when then there's a predator human arrives to a location where the animals haven't seen humans before, they're maybe not as afraid. Now, it might not still been worth fighting a thunderbird or trying to hunt one, but they may have been able to just walk up and take an egg.
The strange thing about the thunderbird and the dodo, though, is that I understand they didn't know humans before, but humans have a lot of telltale signs of predators. They have the binocular vision. And so it's like, we learned on the show about how babies have an innate ingrown fear of snakes and spiders because of evolutionary reasons, because of venom and these sorts of things. A lot of animals have instinctual response to anything that acts like a predator. So why is it that these big, beautiful, dumb birds did not recognize a predator when they saw one?
And they may have, like the dodos, though. The dodos didn't have predators on the island. There were none. But the Australian mainland definitely had predators. Like that, it wasn't a predator-free zone. Sort of interesting. Some of the ways that they were able to identify that this was the bird that they were looking at was the shape of the eggs, the thickness of the shells, but also they were able to pull proteins that were preserved within the shells.
Oh,
So,
cool.
the shell was within the layers of the shell itself. There were proteins captured. Although most of their genetic analysis came back as chicken. Which they believe is contamination and not a relation.
Yeah, most
not
likely.
a close relation to a chicken.
All right, Blair.
Yeah.
What do you want to tell us about, or are you chicken to tell us a story?
No, I have poop for you. Not chicken poop, but polar bear poop from Toronto, from a zoo. And this is a study looking at the accumulation of contaminants in the body. And this specifically is looking at polar bears because they are very good bioaccumulators. They're at the very top of the food chain. They have a lot of fat and they are very good at digesting and eating fat. And that's where a lot of contaminants hang out. So they were a good study species. But this is really generally looking at how to study contaminants in any animal. And so looking at this in the past, looking at contaminants, you would need the animal to be deceased and you would need to check tissue. The way to study biomagnification, really the only way historically, has been through dead tissue. So you can look at the contaminants in there. The method that they developed here was specifically looking at poop, which is something that you can get at any stage of life from lots of different types of animals and learn a lot of things. Our poop tells a big story, no matter how big the poop is.
Poop
if you're a polar bear,
yay!
The poop is quite large. But what they were able to tell, looking at an animal that they knew was going to have high bioaccumulation of contaminants, is they were able to track the amount of polychlorinated biphenyls, PCBs. They are outlawed in pretty much the whole world, but they have a really long latency, so they hang out for a really long time. And so this is something that is important to be looking at polar bears, in other wild animals, and in humans. Because even though this stuff isn't supposed to be used anymore, it's still hanging around in the atmosphere, or not in the atmosphere, in the environment, excuse me. And so the previous studies have linked PCBs in wild polar bears to lower levels of testosterone, which can impact reproduction. And these contaminants can have all sorts of impacts on any types of animals. But what they were able to find out is that because of the way bioaccumulation works, the fact that, like, if a big fish eats a small fish and then a a seal eats a bunch of fish and then a polar bear eats a bunch of seals, that is a exponential growth of the amount of this contaminant into their body, and that was passed along into their feces. So they weren't able to make any large scale declarations based on this study about, what the PCBs are doing, or like what individuals had more PCBs than others, or why that might be. But really, this was just a proof of concept that you could accurately test PCBs and potentially DDT through the digestive system. So this is something that could help with the study of captive animals, wild animals, and humans in the future.
Well, we know that like heavy metals and other things that women who are pregnant are told not to eat too much tuna. And, it's because of this bioaccumulation, and that it would end up accumulating in your tissues, but also being passed along to the fetus. And so, we've known for a while that we can sample poop to learn a whole lot of things. But I think this is, what's interesting here is that they can start actually specifically looking for these really potentially dangerous compounds, long-lasting compounds in the environment, and seeing how they're affecting the food chain.
Yeah. So the poop tells the story, as per usual.
Now I'm replacing Depeche Mode songs with lyrics with poop, and okay, that needs to stop.
Or continue.
Or continue. We'll sing those songs in the after show. Ha. I'll keep that one in my head to myself. You can imagine what I'm thinking, what I'm imagining. Okay, what do we want to talk about next? How about some electric fish? Because I know this conversation is electrifying so far. I'm in a weird mood tonight. I don't know.
As long as you don't electrify the poop,
should I say you're welcome?
You're good. Just
all right, so
the fish, not the poop.
Not the poop. No.
Hey, we're trying to do a tight 90 here, folks. Do you mind if we move along to the next story? I know you've got a lot of jokes you've been trying to work out.
I'm trying
last
here.
Week.
I'm really trying. Okay. Electric fish. How'd they get to be electric?
Oh, that's actually a very interesting question.
They bit into some wires under the water.
They didn't just plug themselves in. That is not
robots
the way it happened.
Okay. Some sort of electrified algae.
Ooh, high iron content in their diet.
No.
Superpower.
Let's go with adaptation and evolution.
We're gonna
go with mutations in particular segments of DNA, kind of like an epigenetic control, where in control regions of genes for a sodium ion channel that's normally in the muscle. It normally has a couple of copies of these in the muscle, and they get turned on. The sodium ion channels are really important to make the muscles contract. But in certain copies, through evolution, these electric fish species managed to, in a couple of different ways, there's a bit of convergent evolution happening as well. They came to the same solution through different pathways along different lineages. But what happens is there's a little 20 or so nucleotide section that is a control segment. And if there get to be mutations in that control segment, it turns it off so that ion channel doesn't make the muscle work anymore. But in different cells in the body, if that is turned off, it leads to electrical ion flow. So the muscles get turned off, this ion channel gets turned off in the muscles, but then these electrocytes in the electric fish get turned on. So it's the same gene, but there's one control segment that either gets removed in some lineages or gets altered so that it basically doesn't work so much anymore, but it works for another function. So no muscle, this ion channel doesn't work in the muscle cells, but it now starts to work in these electrocytes. And the electrocytes allowed the fish then to gain this function. It's just adaptation and evolution. And that's pretty much it. And it's pretty exciting
wild.
That they were able to compare the genetics. They were able to compare a number of different electric fish to actually determine that they're not all the same. It's not just one solution that led to electrification. But there are a couple of different solutions. It's either remove it completely, this little control segment, or just mess it up so it doesn't quite work right. But in effect, it comes to the same solution.
Right. And so there's actually, as phenomenal as that seems, we also have to remember that this electrical impulse thing is, at least in some of them, is used for communication.
Yes.
And so we have echolocation in dolphins and bats. We have speech communication in current modern humans, but we have vocalization in all sorts of vertebrates and birds and birds are vertebrates. All sorts of animals that aren't stuck in the water can use sound, which is a thing that, even some, right, I can see Blair say, even some fish are using clickety clackety soundy stuff to communicate.
But these fish are like zap zap.
What I was going to say is that at least with terrestrial vertebrates, my understanding is that the vocal cords have a shared origin. So I think that's what's different about this is that they're and I guess that's more like the fish you're talking about, Justin, is the vocal cords of terrestrial animals versus the clicks that fish make, which they don't use vocal cords for that at all. So I would say that is a really good comparison in that case.
But I'm wondering if then, like, as this thing evolves along and there's this ability to utilize it, it becomes a communication thing. Because I think of electric fish, all I think of is an electric eel. I think of something that zaps its food to paralysis, right? Like it's a weaponized thing is the first thing I think of. But there's all sorts of interfishy communication stuff that also is going on with that. I don't know.
Right.
I wonder how
they use it for hunting. They use it for defense. They use the electric organ for communication. There are all sorts of behavioral uses that came about once they had this new mutation, right? This new organ that they could then use. So some individuals were able to take advantage of their electrification and use it to their benefit for whichever ways that they could. But it's this, interesting, weird fish genetics that they had 2 different kinds of sodium ion channels, and they had duplicate versions of the same gene so that the muscle cells could do their thing, and then it could silence the others and just be like, oh, you're going to do this other function in other cells over here now. But it's just repurposing, evolutionary repurposing. This happens all the time. We're going to take this, we're going to flip it around this way. It's like an old t-shirt that you hack up and you make into a cute new, into a dress or something. I don't know.
And I mean, I was going to mention fish were the first, big group of vertebrates. And so they've been around for a really, really long time. And they've had the whole ocean to explore for that whole time where adaptive radiation has happened over and over and over again. And so if you want to talk about a place where crazy permutations can happen, I think fish are some of the best examples of that because they've had the benefit of time and diversity of niches and
Tell me about axolotls.
So this
Justin?
is actually absolutely following on that story of of genes being reutilized in different ways. Imagine having the ability to regenerate almost any body part throughout your entire body. All right, a regenerated leg or arm, not a problem. New heart, new lung. Don't wait for a donor. Just regenerate your kidney. Refresh that skin as well and reboot those ovaries if you find the need. Spinal cord got you down. Just regenerate it. And if you don't have a tail now, well, you can. Even reversing brain damage is possible if you're an axolotl. This is a nearly extinct Mexican salamander that has all of these amazing regenerative abilities. Pretty much all that we know of them and study from them is like 150-year-old sample that was taken to Paris and has been built up as a research colony over the years. Animals that have been genetically engineered. It's a very important step in doing much animal research. This is our cloned mice that have knockouts or have been selected for a specific gene trait. They can be genetically altered to have fluorescent tags on some genes, which allows them to be able to be studied to see how cells behave that are connected to certain genes. So all of this is extremely important for research, but there's been few transgenic axolotls available to use in these studies, despite potentially holding the key to regenerating every body part and immortality and the fountain of youth simultaneously, which is important because if you live forever but just continue to get more old and decrepit, it might not be worth it at some point. Yeah. But that's changing. Development of these new tools to work with the axolotl, they say, has now elevated the level of established research models and positioning the community of scientists who use them to a functional research model with exponential growth potential. And following 2 studies that are just published this month in the edition of Developmental Dynamics. And we're only 2 days into this month, so it's very recent. This is quoting Prayag Murawala, a scientist at the MDI Biological Laboratory in Bar Harbor, Maine, saying, with these tools in place, we expect to see exponential growth. We only need to look at other animal models to get a sense of the variety of topics that can be studied. Most axolotl research is now focused on limb or tail regeneration, but there's also the opportunity to study regeneration of the brain, heart, lung, spinal cord, and more. We are not running short on biological questions to investigate. Which actually, that's the one thing that made me sort of like I get the body needs to heal damaged tissue. But what happens if you regenerate a brain? Are you still you? Do you have to fill it all up again if you have like a really bad brain damage? Or do you have to start over? Are you
talking about Theseus's ship right now? I feel like that's what you're talking about.
About what?
Theseus's ship. If you change every plank of the ship over time as it rots.
Yeah, is it still the same ship? Yeah.
Is it a new ship?
Well, no, that's okay. It is because every human body part is like a body part, like a machine, like an arm. You give me a new arm, it's just a new arm. I lose my hand, I get one of those cool Luke Skywalker hands. Not a problem. Yeah, the heart goes bad. Put in a monkey heart with a pacemaker. It's just a part you're replacing, or it's a part that needs to get healed or whatever. But the brain, that's what I'm talking about. That's the thing that I think is me. Right?
Your whole self is you. I mean, it's your brain's interaction. Your brain remembers that your brain has all the networks, but the brain is connected to the body, so it all works together as a system. So if you replace parts and regenerate parts, the brain has to learn how to talk to those again. If you regenerate the brain, the whole brain has to learn stuff all over again. It's always going to have to refigure unless we get to the point where we're just like, choop, scanning brains and choop, uploading consciousness into brain kind of thing, which we definitely are not yet.
The ship of Theseus, by the way, I think is a terrible argument. Look, have you ever had a car? Either of you, have you ever had a car? It's a simple question.
Oh, yeah. I use my car until it is limping.
And you changed parts and it was still your car. And then one day you got rid of that car and you got a different car. And guess what? That was your car. Not a single part from the old car went into the new car, but that was still your car.
That's not the argument.
Enough already. All right. So axolotls, they're going to be the key to our regeneration.
Yeah, and now we have a generation ability to make models with the proper knockouts with those tools, which was lacking which is going to allow research on one of the
faster.
most I think one of the greatest potentials for disease management. Because as we talk about any disease, we're talking about a cascade of negative events that have taken place, and it's causing downstream effects from where the disease starts usually. And if there's a way to trick your body into regenerating the original factory part and rebuild it, that's huge.
Stronger, faster, better. Okay.
Or just as fast, just as strong, but what if, no better than before, and it would still be fine.
But what if you were a clone and your clone just kept cloning and cloning and cloning until you were one clonal organism that was 4,500 years old and lived over 180 kilometers under.
Is this a forest of?
She's talking about the Michael Keaton film Multiplicity, right? Is what she's talking about.
I feel like this is like either some pine forest or some fungal spread.
So not a fungal spread. So we've talked about before the largest organism on the planet. It could be an aspen grove. It could be a fungus here in Oregon. However, Australian researchers have located what is now thought to be the largest plant on Earth. Ooh. And they estimate it at 4,500 years old. And it is seagrass. Oh. It's under about 180 kilometers underwater and all underwater. And they have determined through genetic analysis. First, they were like, oh, let's go find out how many different kinds of seagrass are down here and see how they're evolving. And, blah, blah. And then they're like, oh my goodness, it's just one organism. It's all the same.
And you got me. And you got me right away, because I was immediately, when you said plant, the first thing I thought was like, where could it be that there's never been a fire? Because like some of the oldest growth trees are, because they haven't had a fire in a long time. But still,
and it's seagrass.
Spoken like a landlubber.
They've published their research and proceedings of the Royal Society B on this single plant, a clone of the seagrass Posidonia australis, and this is in the World Heritage Area of Shark Bay in Western Australia. So this is fascinating. They have determined, however, that there are areas within the 180 kilometers that have adaptations and variations. And so its genes do appear to have resilience, and that they are well suited to its local but also variable environmental conditions. And that there are these little subtle genetic differences within the clone that allow it to exist across this range, even though it's a clone. It still has little tiny differences. But now there's more research to go in to determine how the plant survives, how it thrives, and how it really has lived for so long. What made it special? Cloning, really, but
cloning, but also I think going back to the plant living without having to encounter fire, a relatively consistent biome. You know, if the environment that your niche really works in doesn't change for 4,500 years.
Yeah, but that's the thing. It has changed in 4,500 years, and it's managed to survive. And so what they are saying is that it has these subtle genetic variations, and also this variability has allowed it to be resilient in the face of change. And that's what they are going to investigate further.
See, this is your real ship of Theseus, because as it clones, at what point is it no longer the same individual? If it has cloned itself and there's slight genetic changes over time?
That's what humans are, isn't it? I mean, that's our same thing.
We don't clone ourselves.
Okay. So it's not one individual becoming another one individual. It's two becoming one, which is
but there's
not that far away.
It's a huge. There's a There's an almost infinite number of different combinations that you can have in all of your different, kind of matchings of chromosomes. And in this case, it is like 99.9% exactly the same, except there's a slight difference. So
right.
Well, that's just because we're used to thinking that's the complicated way because we're Earthlings. On Omicron Perseus 3, it takes 30 to have one baby. 30 have to get together to make one offspring, and it's a mix of all of those genes. So that's
there's a novel, maybe it's Vonnegut, I don't know, but there's a novel somewhere where there's extra dimensional beings that actually are part of human recombination mating that we don't know about because we can only see.
I think that's
no.
I think that's the best.
I think it might be a Vonnegut.
I don't want anybody else involved in that except the child. In the end, you're done. Okay, go grow, be a thing. All right, we have finished our first set of stories. Thank you for joining us for this weekly episode of This Week in Science. We hope you're enjoying the show so far. If you are, please tell a friend today to listen to TWIS. All right, let's do a few COVID stories. You want to talk about COVID? COVID!
Not really.
I suppose.
The story we don't want to talk about. Okay, so this one's it's COVID related because of its impact on our health moving forward. So the pandemic has led to people in isolation. We have been wearing masks. People have been social distancing. We've been washing hands and doing all sorts of things that have reduced other illnesses, flu, respiratory syncytial virus. The cases have gone down, down, down. And then what we've seen is that there's a new study, it just published in Nature Communications on off-season respiratory syncytial virus infections in Australia after the easing of COVID-19 restrictions, which led to off-season. So if these colds are usually winter colds, that's when we normally get these rhinoviruses, these syncytial viruses. They're now in the spring and the summer and not during periods of time when we normally get sick. And so the circulation of the viruses are different. And in fact, with genetic sequencing, they revealed that there was a major reduction in the RSV genetic diversity following COVID-19 emergence, with 2 genetically distinct clades circulating cryptically. So they were like hiding in there, probably localized in very small regions before an epidemic surge came out after the COVID-19 control situation was relaxed. They say there also is the possibility of some viruses maybe going extinct. And
that would be nice.
But there are others that are still circulating. So of course there's going to be new cold viruses and other things, but it just highlights the need for continuing surveillance and sequencing of these other viruses. We've also seen hepatitis that has killed some children in the United States and around the world that we don't know the cause of the hepatitis, and it's thought to be related to an adenovirus. And an adenovirus that would normally not be a problem, but that potentially is more dangerous because of the lack of exposure in the previous years, or because of the immune system kind of losing its responses over the last couple of years. So there are some very interesting questions now as to how, as we get through these pandemic waves to come, as we, slowly over the next couple of years kind of make our way out of this pandemic, what will be happening with other viruses that we normally see as seasonal? Will COVID itself become seasonal? Will these others become sporadic and difficult to predict? Will winter colds come back and just be winter colds again? And how long is it going to take before the ripples of all of these experiences end up becoming a still pond again?
I'm curious if there are people, especially children, that did not get their vaccines on time
for sure.
Other things.
Absolutely.
Because
yes.
people were very concerned about going to medical facilities. People were told to put it off during surges. Now it's really difficult to get appointments because everyone's starting to go back. I'm really curious to look back at that because I think it has to have impacted vaccine schedules for children for things besides COVID.
Yep. Absolutely. So there are going to be some interesting just public health ramifications over the next decade or so, not just from directly related to COVID, but also these kind of off-target effects of the whole pandemic and what's happened. Yeah.
I just got my Tdap booster after 10 years. I got the notification.
What?
Oh man, really? It's been 10 years already?
I don't know that I've ever gotten I don't even know what the Tdap is.
You probably got a Tdap. You had a child.
Tetanus, diphtheria.
Oh, tetanus shot. I've gotten that one
something
before.
Tdap. They put it all together.
Pallidum. I don't know. I'm not a doctor.
Okay.
As we are going back through COVID waves, we know that one of the places, or one type of place that has been a problem for infections has been the gym. People going and working out, breathing very heavily. But there's been a question as to exactly how much aerosol is produced by different individuals during the course of exercise. And so some researchers decided to throw a bunch of people on treadmills and measure the aerosols that they produce.
Don't tell me the answer.
Okay.
Because I have been during this whole pandemic, we'll be out in public, and once you're out in the free open air, you take off the mask, and you're like, I can breathe without a mask, not close to humans. And then some jogger comes by, and you're like, oh God, why did you have to run by me? Because you're breathing heavily. But then I'm also thinking, if somebody's doing heavy breathing all the time, haven't they kicked out enough virus, if they had it even, that there would be less?
I don't think it works that way. I don't think it works that way. It doesn't work that way.
They used it up, right?
Well, or was it?
I just blew it all out.
I mean, is there not that it would be gone, but is it less than somebody who wasn't going for a jog and breathing on you?
Well,
anyway.
what they determined that for untrained people, the respiratory volume increases from 5 to 15 liters per minute at rest to over 100 liters per minute when exercising.
Ooh.
And highly trained athletes, because they have a higher volume of oxygen that they take in, that actually reaches up to 200 liters per minute. Yes. And then, and so this is respiratory volume, and what they've correlated that to, of course, is the amount of aerosols that are expelled during exercise. So if it's kind of exercise that really, a little light jog, you're not really well trained, you're going for a walk or maybe a little jog, doesn't really get you breathing that much, it's not a big deal. If you're going for it and you're huffing, puffing, breathing, that huffing, puffing, breathing is producing a lot more aerosols. And the reason that this in particular is important is because as we go through waves of increased COVID transmission, it's good for gyms to know, okay, people are working out really hard in our gym, so we need to open the doors. We need to increase ventilation, or we need to take all of this. We do HIIT training, or boot camp or whatever. Go outside and work out outside where there is a lot more air. So it can help protect people. If people who are younger, fitter, and they're in the gym, they can wear a mask to protect others from their increased aerosol expulsion if they are going to be working out at a higher rate. So it helps gyms make decisions. It helps public health in professionals make decisions.
I can give a couple of examples. Both the treadmill and the exercise bike are simulations of an activity you can actually do outside. Outside. That's how those started. And then somebody said, we can't charge for that. So they created a way to do it inside. So you could have air conditioning.
There's also places where like
handy.
your sneakers would melt if you went for a run
outside.
Yeah. So it's there are
Pakistan's certain things. Like 120 degrees right now.
Yeah. There's certain things that you can't do outside all the time.
It's true, but these things are important for public health and to maintain things. Hey, Blair.
Yeah.
Did you get a chance to look at the update for the COVID sniffing dogs?
Yeah, I did. And they're good at it.
Which is what we said before.
Yeah, so this is, I think, the third time. I tried to find the other stories that we reported on before. I had trouble finding it. But the latest one was back in December, where they found that dogs were better than antigen tests at sniffing out COVID.
This is also the story that I've never believed.
So in this one, let's talk about that for a second.
Yeah.
This one was looking at samples from 335 people. They trained dogs to sniff out coronavirus cases based on sweat or other bodily fluids that were put, into a container, and then they smelled a cone that kind of directed that smell into one place. And so they were sniffed out 97% of coronavirus cases that had been identified by PCR tests. And they found all 31 COVID cases among 192 asymptomatic individuals. So,
wow.
Yeah, so they were actually very good at detecting asymptomatic individuals, which is exactly where dogs could come in handy in places like airports, concerts, sports events, because the people who feel well are going to come. And so the dogs could sniff them out and they could be pulled aside. And so this is part of the thing, Justin, is even though this article is suggesting that they're actually as or more effective than the tests that we use, this could be used to identify an individual to then take a rapid test and then be identified what to do based on that. Because no matter what the science is telling us, people are still very dubious of dogs in this regard. Now, one of the researchers mentioned that people don't think of dogs as high tech the way electronic sensors are, but they argue that dogs are one of the highest tech devices we have. They're biological sensors instead of electronic sensors. And if you trust a dog to sniff out bombs or drugs, why would you not allow a dog to sniff out COVID? It's the exact same chemical processes.
I'd love to know, what subtle shift in our aroma gives it away. Like, what is it? Like, what changes in our sweat or just... That to me is so fascinating.
Yeah,
what is
and this is what we don't know. And part of the crazy thing is you can use odor from body sites at different locations on a body, different types of odors, and all of them can give this information. So it doesn't appear to be a single hormone or a single chemical. It really looks like a pattern of increasing or decreasing certain smells in a person who is carrying a disease. Because also remember that body odor is completely dependent on genetics, what kind of cleaning solutions you use, how often you shower or change your clothes, all sorts of crazy things that can impact your particular flavor, I'll say. But the dogs can still tell if your flavor is off and you are therefore carrying COVID-19. So this really is looking like these dogs are extremely effective, and they hope to start using them in these larger arenas. Some, since the December research that we reported on, have been used at concerts and large outdoor events to great success so far. But the real problem is that there's actually a supply chain issue with dogs. There's not enough dogs to do what they currently do, detecting explosives and drugs. There are so now to get them to detect diseases is a whole extra problem. So the issue is, we have lots of dogs in the United States, lots of dogs, too many dogs. Spay and neuter your pets, but ultimately, the problem is in training. It takes a lot of time and care to train dogs. Not every dog that starts training finishes training. Not every dog that is successfully trained in a closed scenario can then perform
public.
in a public space.
So because this is one of the things too, whenever I look at this, is the drug sniffing dogs. I get they can absolutely do that, but then the ones that are being deployed by law enforcement across the United States are worse than a coin flip. Apparently, like they're actually pretty bad at it, and a lot of their
Real
indicators that we don't see are false hits because the police officer has a bias and wants to have a reason to search a car.
Yes.
And then they don't find anything, but it doesn't end up in the report. So like there's always, it's training, but like you said, even if they got trained out into the real world, how well
and that's
is that
the other
maintained?
piece, training the handlers, right?
Yeah.
And so if you have dogs that are part of a organization where the handlers are also trained and deployed and part of a specific group, then it can be easier to kind of do this. So there's actually there's organizations that take rescue dogs, for example, train them to do a job like smell out invasive species, and then the organization with their rescue dogs are sent out to areas to work. So this is what the dogs that I was talking about that worked at the outdoor concert earlier this year, this is how they worked. They were from a company. So it's tough. It's harder when you include law enforcement, because then you have somebody whose their main job is not trained to be a dog trainer. Their main job is in law enforcement. This is another piece of that job, but it muddies the waters. So this is definitely something where there's a lot of variables, just like there's a lot of variables when you have a sensor that you could be using or not using correctly, right? So it's not a perfect system, but these dogs seem to be very effective at smelling COVID.
I feel like it's also going to be the consistency of the work, because I feel like airport sniffer dogs are probably getting a high volume of traffic and are probably catching 3 or 4 people or isolating several, probably dozen pieces of luggage a day, if not an hour, for traces of stuff, right? But then you go out, like, you got the dog that you're going to use for the event, but there's not an event for the next month. And so you do some training, but then you're like, oh, he looks hungry, I'll give him a bone. Anyway, suddenly the training, like, so, you know.
Justin!
Oh my God.
It's the consistency of keeping that training as part of the behavioral reward system.
Which they usually do with these animals.
They
try
to.
They would do.
They try to.
Like an
They would
airport
do.
Yeah.
dog, I see
But-
working all day long.
That would be
Any
the
other
whole
scenario,
point,
I feel
and
like
they
it's
would do
the
that,
best.
and that's what you pay them for. Yes.
You know what?
We pay the dogs?
No. You know what? This is This Week in Science. Thank you so much for joining us. And you could pay us to do this if you like us that much. If you like TWIS, head over to twis.org, click on our Patreon link, and you too could be a supporter of TWIS. We are listener supported, so your donations monthly, whatever amount through Patreon, allow us to do this show for you. We really appreciate your support. We can't do it without you. Let's come on back right now for that lovely part of the show known as Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I have baboons.
Yikes.
Yes, baboons with special male friendships. Yes. So baboons, specifically guinea baboons, live in really interesting societies called tolerant multi-level societies. They have core units consisting of 1 male and 1 to 6 females, and they're young, so far sounding familiar in terms of many things that we've heard of in the animal kingdom. 1 male, many females, and the young. Several of these core units all form together with some bachelor males that have formed their own party. 2 or 3 parties team up as a gang, and then all the offspring are fathered by the main male representative that is the core of the unit, while bachelor males hang out but are not sexually active. So they have this big society formed of these smaller parties, which are formed of smaller core units. Confused? That's fine. Just understand there is a male with a bunch of females that are essentially they're his for a mating season. They live adjacent to other similar groups, and there are bachelor males hanging out in the area. Hopefully that makes more sense.
It's good to be the top baboon male.
One of them. Yes, there would be a bunch in the area kind of saying, hey, sup, but staying in their own spot, right? So what this study is looking at from the German Primate Center, the DPZ, which I'm guessing is the Deutschland Primate Center. I don't know. Anyway, they wanted to look at the bonds between males because they seem to have a really strong bond with one another with no rank hierarchy when these males form these friendships. So why would they do this? Does this help their mating success? Does this help their survival? Why do they have these male friendships when ultimately there is going to be one male that is in charge of an entire group of females? So how is there a benefit here? This is something you don't often see in groups where you have one male and a bunch of females. So what they found was that there was no benefit to male reproductive success in having male friends. In fact...
That tracks.
Sure, yeah. In fact, once the males associated with females and began to have babies, they spent less and less time with their buddies.
Yeah.
Right?
Yeah, that tracks.
Can relate. Although males in their prime maintained some social contact with other males, they adjusted their investment of social time according to their life history stage so that they could maximize reproductive success, which meant paying attention to the females and their own babies. So there's a bunch of theories. Do these male friends provide emotional support? Do they help in defense of females against rivals if another core male is trying to come over? If you're friendly with some bachelor males, will they help protect the other females that you have? Difficult to say. One thing we know for sure, it does not make them more likely to have babies. So one of the ideas that might be happening is that this just allows them to hang out near these other animals, which allows them overall fitness success because they're not off on their own. They're not going to go die by themselves, right? So basically just being friendly with one another allows them to be part of this larger, looser social group. They looked at the social behavior of 30 males, determined the paternity of 50 infants over 4 years. They looked at over 400 individuals in total, starting in 2010, and there was no reproductive impact. There was no evidence that males with friends were more attractive to females. It wasn't that they were getting paid more attention to. So really just young and old bachelors have time on their hands. And they hang out with each other because they don't have females to hang out with. So
and maybe they've got buddies and it keeps them from being in trouble. Like,
right.
Oh, here comes a predator, but now there are more of us.
Yes.
Or, oh, we've got more access to food. And yeah.
So
buddies
it really just
good.
feels like...
I feel like it's more like, the females don't want to hang out with me for some reason. Hey, me too. Oh, okay. Well, I guess we have something in common.
So it's definitely a function of this strange society shape that these baboons have is it allows for, in gorillas, for example, a lot of the time you'll see bachelor troops. So you'll see just a bunch of males that aren't paired off all together by themselves, not near females, not near successful males. They just hang out and do that for a while until they can find a place to go to take over. Similar with a lot of hoof stock, lots of, giraffes and antelopes and stuff like that will do this too. They'll have their little bachelor groups. But in this case, because they have this larger, more tolerant social structure, they're allowed to hang on. And because of that, it allows them to have this very funny interaction where they have this friendship with other males, and then all of a sudden they start to become the big man on campus and they don't have time for their buddies anymore.
Oh,
yeah.
And the buddies are like, oh, he never has time for us anymore.
Never has time for He never picks the bugs out of my fur anymore. I really miss him.
Aww,
What's up with that guy? So next steps, maybe when he gets older and he gets ousted. Next steps for this study is to look at whether male friendships help to attract first females earlier or maintain status as a reproductive active male for longer. So while as they haven't found statistical significance with the number of offspring or mating success, these are the next measures they want to look at. Does hanging out with men and having friends and social interactions, does that make them better at interacting with females? Does it help them to hold on to their leadership role for longer? So these are things that they can look at.
Gotta say, if it's anything like humans, the answer to whether or not hanging out with their buddies makes them better at attracting women is gonna be a no.
Unless their friends are real allies, really evolved individuals,
evolved baboons,
intuitive.
Yeah.
Hey, I heard, a thing I heard about what the ladies like. Oh, what's that? Yeah, apparently they're into when you bathe. Oh, that's weird. Yeah, who's ever heard of a thing like bathing before? Yeah, no, I know, but that's apparently what the ladies like. Oh gosh, wonder if we should try that. Nah.
You know what females really like, Justin?
What?
Females really like it when you move your pedipalps. When you have your legs, you tap them, and then you drum your forelegs with really quick speed. And then you have baseline tremors of your abdomen. You slap your abdomen on the ground a bunch, and then transmit that sound, those baseline tremors through your 8 legs into the ground. Make the ground shake, and then eventually make the female shake.
Hey, I've heard another thing about what ladies like. Oh yeah, what's that? Well, it's gonna take 4 of us.
Yeah.
Or
you could be a wolf spider.
Oh yeah.
So for wolf spiders, some of the secret of courtship is simply having the most complicated dance. This is a study looking at male Schizocosa stridulans spiders, which are a type of wolf spider, and what makes a successful dance. So in this study, they deposited a female onto filter paper of a soundproof chamber a few minutes before they introduced the male. Then the team recorded ensuing courtship with the help of a camera and laser vibrometer.
I love that. That method is so cool.
Yes. I want to hear, I want to see, I want to feel. By shining the laser down at a piece of reflective tape stuck onto the filter paper, they were able to analyze vibration-driven changes in the frequency and other signatures of the reflected beam so that they could capture every little bit of signal from a male as he danced. What they found was that the more complicated, the more vim and vigor in the dance, the more successful the performance. These could last 5 minutes or up to 45 minutes. Can you imagine watching a spider dance for 45 minutes? I mean, I could. I definitely could. Get me the popcorn.
Actually, I think I would be down.
But I also watch frog videos for several hours at a time, so
I've gone to a modern dance concert or performance before, which lasted, I think, for I feel like it lasted like 8 hours.
Yeah, I'd rather watch a spider.
Yeah, me
I
too.
think, than that. Yeah.
This sounds like a modern dance performance.
That's on point, which is much harder than it
That
looks.
was like tap ballet.
Yeah.
How do you lace up 8 little bitty ballet shoes?
Amazing.
Well, anyway, they looked at 44 of these performances. 9 of the males were awarded with copulation. They also had the most complex courtship signals of the bunch. And this was held across 3 different measures of complexity. These measures were previously used, you gotta love science, to characterize binary code, bird song, and whale sounds. There were no previous models to study the vibratory signals of arachnids and their associated complexity. Yes, so this was a very interesting way to turn this just like, wow, that looks like a wild dance, into actual like math and numbers and something measurable that you could then do something with, do a science with. So so there's a couple theories as to why more complex signals would be the desired trait. We ultimately don't know. We're not in the female wolf spider's glob of neurons that they have. It's not quite a fully, it's brain. The
glomeruli.
Yes. But one, the most common answer based on a lot of Darwinian theories would be the more complex signal takes energy and time to produce, and therefore that is a signal that they are more fit. They are more doing something harder and something that would actually call attention to predators and not dying means you are the cream of the crop and you should be allowed to mate. That is the very, very baseline, like
the law of the jungle. Answer. If you can make a lot of noise in the jungle and not die, then you're great.
This is peacocks.
You're the best.
You got a giant tail on you that makes it harder to fly and slow. So if you have a huge tail and you haven't been eaten, there's something special about you. But the other idea is simply that females are bored. They're so bored of it. They just see the same thing over and over and over. And so anything novel would be something that they would be interested in. So this is, increasing complexity, especially through time, is as if the males are changing things to keep their interest. So the females are like, yawn, snore, same thing over the, what is he doing? This is a very weird dance and sound. I've never seen this before. So
next thing we know, the spiders are going to be like mockingbirds, and they're going to be making car alarm sounds. And
you know what I was thinking about actually was about the fashion industry and how when you see the runway, you see the absolutely crazy fashion choices that designers make, and you're just like, what is this? It's my mom used to always say to me, and I was like, what is with this new stuff? She would say, they're just trying to be different. Like, different is eye-catching. Different is interesting. And so just try it's very smart, right? So that's exactly what this is. It's in humans, we do our own version of peacocking or spider dancing, where we're trying to stand out in the crowd. We're trying to do something different and daring. And so maybe that's all this is they're just trying to move around and look crazy enough to get lucky.
Somebody notice me.
See this crazy thing I'm doing? Look what I can do.
Doesn't work out for a lot of kids. Look what I can do. All right. Moving on. What can you do, Justin? What stories do you have?
There's a couple of stories that I still have to do to talk about. But my computer's slow. There is a 3,400-year-old city under the waters of Kurdistan that has been revealed thanks to climate change.
Atlantis?
Climate change is revealing all sorts of stuff. There's all sorts of ice patches that are melting, and there's artifacts coming out of the flows. This one is thanks to a drought. Major reservoirs saw its water levels drop like a rock due to an extreme drought in Iraq, and the waters receded enough to show that an ancient city was underneath. It had a palace, there was massive fortifications with walls and towers, a multi-storied storage building, and an extensive urban and industrial complex were attached to this thing as well. So a team of German and Kurdish archaeologists raced in to document as much as they possibly could of this emerging city before the waters returned, and that was really their timeline. The waters have exposed this city under a water reservoir, and it's going to rain again, and the reservoir is going to rise again. So here's your timeline now until when the thing fills up again. So working under this pressure, the researchers mapped out the city, and then they actually started getting some pretty insane finds from their excavations. So there were these amazingly well-preserved walls. So these were several meters high, which is not bad because these are sun-dried mud bricks that have been underwater for about 40 years now. Part of the good preservation is because the tops of these walls had collapsed and covered buildings, and there was like this earthquake that happened 3,000 years ago that caused an earthquake for the tops of these buildings to fall. So you have these buildings underwater covered by other structures, other earth. The most amazing thing that they found, there were ceramic vessels with over 100 cuneiform tablets. These are sort of hieroglyphic language tablets. These date to the late Middle Assyrian period, shortly after the earthquake. So that's about 1350 BC. So you've got written texts over 3,000 years old, written in hardened clay, and they were enveloped in a soft clay, because apparently that's how you used to send letters. You would take your hard clay thing that you baked in the sun, and then you'd take soft clay around it to hide what it said, and then you'd hand it to the postman or whoever who would deliver, and then whoever got it would like peel off all the soft clay to see the letter beneath. So they got 100 of these. And then those
obviously weren't writing on paper yet.
They didn't have the paper there, at least not here in Kurdistan.
Yeah.
So then these are in these vessels. They got stored in there. Then they were in a building that then got buried by a wall that then was underwater for 40 years. So it's amazing that they're now in the hands of archaeologists. The rising waters have returned. The excavated buildings. They were complete. They were covered with this sort of tight-fitting plastics. They poured gravel on top of that to hold it down, and they're now waiting for probably not too far off in the future time when they're able to get back to the site and do some more digging. But they've got hundreds of these tablets that they can now decipher, and they're likely very domestic communications. Mm-hmm. Which is probably going to give a very good indication of daily life, because these are sort of randomly taken from households and storage areas. So they might be business transactions about goods coming in and out. They could be interpersonal communications. It's going to be really fascinating to see what the results of these are. And
that'll be wonderful, amazing. Thanks for that story.
Halfway around the world. A network of lost ancient cities in the Amazon was discovered by flying lasers. This is entire cities and networks of cities under centuries of wild growth, hidden by beneath thick tree canopies in the forests of Bolivia. These cities, built between 500 and 1400 AD, had elaborate, intricate structures, unlike anything that they've discovered in the Amazon to date. These are 5-meter high terraces. They say it's covering 22 hectares. I have no idea what a hectare is, but apparently, according to this, it's equivalent to 30 football pitches. Don't know what a football pitch is, but if it's anything like a football field, soccer, like 30 soccer fields. There was also 21-meter tall conical pyramids that were discovered. So this is also how intense the forest canopy is to hide these things. These are extremely difficult areas to access for an archaeological survey by foot. This is the machete all day long, and then you've gone 10 yards into this forest kind of a thing. So they flew LiDAR detectors, which is basically laser stuff that shoots through ignores all of the foliage and shows the hard structures beneath. And it showed this amazing, intricate, they found vast networks of reservoirs, causeways, checkpoints spanning several kilometers. And this is, let me find it, 600 miles of canals along raised causeways connecting sites to reservoirs and lakes. This is a much more intricate water delivery system than California currently has. Just really amazing. Research is published in the journal Nature. The LiDAR technology that blazes in the sky are able to peer through the tropical forest canopy and examine the sites found in the savanna forest in the South Amazon. This is an area at the same time that they say these cities were being built and these networks and all this water infrastructure was being built. The seasonally flooded Amazon savannas were being transformed into productive agricultural land in a region that is roughly the size of 90 Rhode Islands, as we Americans would put it, or as the British would say, one jolly old England worth of territory dedicated to agriculture in this method. So, yeah, co-author Mark Robinson, University of Exeter, adds, our results put to rest arguments that Western Amazonia was sparsely populated in pre-Hispanic times. Absolutely. It sounds actually like one of the biggest agricultural sites on the planet at that time.
So how old is this again?
This is from 500 to about 1500 AD.
Okay.
Is when it was operating. So yeah, about 1500 years old is when it began. It was built.
Yeah, I was just trying to place it in terms of like Roman aqueducts and other, technology. Like you're making fun of California right now, but in ancient times, we've been pumping water for a while, and the Romans get all of the glory for their water systems.
The other things that they sort of point out in this, that the cities and the way that they were designed, constructed, and managed was very much with the natural landscape in mind. So they employed there was sustainable subsistence strategies. There was conserving nature areas within the living areas of the people who are living in those urban landscapes, so that there was no point anywhere in how they designed their city where you were completely isolated from access to nature, which is what you used for subsistence. They're very different than the current modern city design, where let's keep nature completely out of the city despite still relying on it entirely for our subsistence.
Right. We'll bring it in. It's part of who we are. Yeah, it's all of it. That's so cool. And I love also the fact that it probably allowed much larger populations to survive than we previously considered. These kinds of findings make you start thinking so much more deeply the where and the when and the how. And hey, Blair, when do you exercise? Like what time of day?
At 5:15 in the morning.
5:15 in the morning. Okay. Yeah. Justin, do you exercise?
What? Why? Just getting out of bed.
It's exercise.
That's about all the heavy lifting I'm going to do.
Yeah. I used to, when I was young, my exercise was all at night because it was had to be after school. So it was like evening, 5 to 8 p.m. kind of exercise time. College was that a lot also. It was like the only time in the evening when I could finally make it happen because I'm not a morning person, unlike Blair. But there's this question about the ideal exercise time of day. Is there a best time of day to exercise? Well, according to a new study in Frontiers in Physiology, researchers took a group that wasn't as big as I think it should have been.
As is usually the case with human studies.
Exactly. Mm-hmm. They started with a total of 140 women and 63 men from Sarasota Springs, New York, who responded through emails, flyers, and local newspapers to advertisements regarding their exercise study. Ended up with 59 women and 30 men being screened and finding 30 females and 26 males who ended up being eligible for participation in this particular study, in which they split the groups in half, and they had half the group do morning workouts and half the group do nighttime workouts. All the workouts were assigned and they were exactly the same. All of the people had to be a fairly fit level of baseline fitness so that it wasn't like, oh, we've got a mix of different fitness levels. It was just, let's get that control factor taken care of already. And 12 weeks later, they had the results of the study. What they found is that while both times are good, exercise is good regardless. Mm-hmm. Morning exercise.
I'm still waiting for the study. It's like, turns out exercise actually bad for you. You should stop.
That hasn't come out yet. That's not the one that we're talking about here. Nope.
But I love that they still point out every time. Any level of exercise is better than not.
Please just get off the couch. That's what they're saying.
Pretty much. Or even if you're on your couch, lift your feet if you can. Do something.
Do a couple ankle lifts.
If you can. Yes. Move your ankles, roll your wrists, stretch. Morning exercise reduced abdominal fat and blood pressure in women. And in the evenings, the women who worked out in the evenings had enhanced muscular performance, but didn't have the same reductions in blood pressure or abdominal fat. So morning is good for bellies and blood. Evenings is good for the muscles, according to this study. In the men, evening exercise increased fat oxidation and reduced systolic blood pressure and fatigue. And so in this also, when there is a particular aspect of benefit to mood, the evening exercise didn't have the same benefit to mood that morning exercise does.
Well, I guess that makes sense because you go to bed right after. You get to do your muscle recovery, so the muscles that you broke down,
the physiology of this might make a lot of sense.
It all kind of makes sense,
Yeah, except this wasn't seen in men. This was not the same. It wasn't the same result in men. So women had this split result.
Yeah.
Morning exercise had a different result than evening. Men, they just found that this evening exercise increased fat oxidation, increased systolic blood pressure, and or reduced it and reduced fatigue. So the key to this study is that there were a lot of factors that they couldn't control and that they didn't control. And they know that, and it wasn't a really big study. So we have a very limited sample size, which could also impact the results because you also have women's menstrual cycles, and there were also peri and postmenopausal women in the study as well.
That was one thing I was going to ask. Yeah.
Yeah. So there's a lot of stuff in there, especially on the women's side for that. And there could be a lot of things to explain the results, but what they suggest is that this result could be used to optimize your individual exercise health benefits. So if you're looking for reduced blood pressure, then or exercise
out in the morning,
or in the evening for men, or the afternoon. And so so exercise, it's good, but the exact time of day might be different for different people and for different reasons.
And of course, I read this story to Brian last night, and as soon as I was done reading it, he goes, "So what does that mean for me? Because I work nights." I was like, I don't know.
It's all messed up with that circadian rhythm.
Yeah, it's because who knows if it's that, or if it's about when
you're eating,
light out, or when you're eating, or when I don't know. I don't know.
But he also this is also like a whole thing anyway, but like a whole separate thing. But like he has a job that is not sit arounding.
Right.
Yeah. So like there's if you have a job where you're on your feet all day, you probably don't need to go to the gym to get the cardio stuff happening. If you have a job where you sit all day long, you probably really need to do one of the treadmill things.
One of the two. Morning or evening.
5:15.
Yikes.
Or get one at your desk.
8:30?
I don't understand how come people don't have like in place unicycle that they can type at.
Oh yeah.
Do something if you're gonna have to sit there all day.
Yeah. Jeez. Oh, but what about what happens when you take antibiotics? Have you ever taken antibiotics and just not only had it affect your appetite, but maybe it made you feel a bit tired and just not energetic?
Yeah, but you always wonder, is it because you're taking antibiotics because you're sick? So is it the sickness? Or is it the antibiotics?
Exactly. Well, some researchers just published a study in the journal Behavioral Processes in which they weren't looking at people, unfortunately, but they decided to look at what happens with gut bacteria in mice after 10 days of antibiotics. And so they believed that an animal's collection of gut bacteria, its microbiome, would affect digestive processes and muscle function, as well as motivation for various behaviors, including exercise. And dun dun dun, the study reinforces their hypothesis. So they found in their groups of mice, they separated them out. One group of mice was bred for high levels of running activity, so they were very active mice, and some mice were not. So they're a little like more normal sedentary mice. Neither had any signs of sickness. So they were all very healthy. And then they were given the antibiotic treatment. Again, no signs of sickness. They seemed all to be fine. The athletic mice suddenly stopped. Well, they didn't stop running, but they reduced their activity in the running wheel by 21%. Just out of nowhere. The high runner mice didn't recover their running behavior for 12 days after the antibiotic treatment was given. So there was and in mice lifetime, that's like 2 months because their metabolism is so much faster. But the way that they think about this is that a casual exerciser, and additionally, the behavior of the normal mice wasn't affected as much during either treatment. So normal mice, they did reduce their activity a little bit, but not to the extent that the athlete mice did, the runner mice. And so what they think this implies is that normal people going about their lives, if you don't consider yourself an athlete, you're probably not going to be affected too much by antibiotics and the effect on your microbiome. But they suggest that potentially world-class athletes might have much larger setbacks from a course of antibiotics for an infection that could really affect their ability to compete and perform. But we don't know because we haven't looked at it in people. It's just in mice. Yeah.
Yeah, it makes sense. The difference between competing elite athletes, if you're talking 2-3% difference, even like it becomes significant. Whereas the difference of 3% and 2 couch potatoes is still 2 couch potatoes. It's how it's gonna break down. Is what I'm trying to say. It's like volume and percentage things are relatable.
Yeah. And in this, what they want to do moving forward is pretty much figure out what the metabolic signals are. So the microbes are in there. What are they doing? What are they breaking down? What metabolites are getting sent into the bloodstream, or what nutrients are not making it into the bloodstream because the microbes are dead and not in there? What aspects are there, and is there the possibility of pinpointing specific bacteria for increasing athletic performance? And this is not specifically to enhance athletic performance of athletes, which you would think might be interesting. They're suggesting enhancing performance of for people who can't move, or if there are risk factors of, if you don't exercise, risk factors for mental health, depression, physical health, all sorts of other things. But if people who are unable to exercise as much, but you can get benefits of good bacteria.
That respiratory rate though of the pro athletes from I think the first story tonight.
Yeah, the 200
right?
liters per minute. Yeah.
In that, you're also talking about a transport of nutrients that's going to be much higher. Yeah. And so that not having the conversion rates taking place in the gut that you usually rely on to feed that transport system, to feed muscles and feed energy levels and all this stuff that's going on. Yeah, that can be severely impactful. I still question how much it will affect those who are not how it would like this study said, not much of a difference in people who aren't exercising a lot. But I'm not sure that that's the best route to.
But there's the extremes, right? So if you have a bell curve or distribution, then you have people who are the average. It's not going to change or affect much, but the people on either end, any changes could be very significant to their performance.
So this also reminds me of another story which I didn't bring tonight, so I will mention very briefly, but it's related to this. It's that in a survey of, I looked it up, 551 primary care clinicians with a bunch of hypothetical scenarios of when they would or would not administer antibiotics, 71% of them in the survey indicated they would prescribe antibiotics to someone where they have bacteria present in their urine, but there are no symptoms to suggest an infection. So 71% of these respondents would give antibiotics to asymptomatic individuals. So it's
just first line of defense.
Relevant,
just kill the bacteria.
Even though that is a counterindication, it's not counterindication. It is against the recommendation of when to prescribe antibiotics, not just because of antibiotic resistant bacteria, and that's its own huge kind of sword of Damocles over us for the future. I don't know what's going to happen when our antibiotics don't work anymore, but aside from that, this could have other impacts on someone's body. If you're prescribing antibiotics when they have an infection but there's no bad symptoms, you could put them on antibiotics just in case and cause all sorts of other problems.
We don't want to cause more problems. No. We want to cause fewer problems.
Yes.
Yeah. And we want to give you as many directions to find your answers as possible here on This Week in Science. Have we made it to the end?
I think we did. I think that was all of the stories that I had to talk about anyway.
I think that's all my stories for tonight, too.
I have one. Oh, no, I'm kidding. That's it.
Oh, Blair. You always kid.
They sequenced the genome of people in Pompeii.
They did.
How did they do that? Well, they did. You should look that one up.
Look it up.
All right. Thank you, everyone, for listening. We hope that you enjoyed the show. Let's give some shout outs To Fada, thank you so much for helping with show notes, show descriptions over on YouTube, social media. Very helpful over there. Gord, Lore, others, thank you for manning the chat rooms and being there to make sure everything flows nicely and people are kind to each other. Identity 4, thank you for recording the show. And Rachel, thank you for editing and for your assistance. And to our Patreon sponsors, thank you for all of your support. Thank you to Teresa Smith, James Schover, Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tozzi, Woody MS, Chris Wozniak, Dave Bunn, Vegard Chefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Gaurav Sharma, Ragan, Derek Schmidt, Don Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Mac Bass, Vote Beto for Texas, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard Brendan Minish, Johnny Gridley, Remy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Philip Shane, Kurt Larson, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Peckerauer, Tony Steele, and Jason Roberts. Thank you all for your support on Patreon. And if you would like to support us on Patreon, please head over to TWIS.org and click on that Patreon link. On next week's show,
we will be back Wednesday, 8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels and from TWIS.org/live.
Do you want to listen to us as a podcast? Maybe while you reorganize your closet? Just search for This Week in Science wherever podcasts are found. If you enjoyed your show, get your friends to subscribe. If you enjoyed your show, this show we do for you, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes, links to the stories we referenced will be available on our website, www.twis.org. You can also sign up for a newsletter maybe occasionally sometime.
It'll pop in there when you least suspect it. You can contact us directly. Email Kirsten at KirstenThisWeekInScience.com, Justin at TWISminion@gmail.com, or me, Blair, at BlairBaz@TWIS.org. Just be sure to put TWIS, TWIS, in the subject line. It will be biomagnified into many trophic layers until it is pooped out by a polar bear in Toronto and looked at by a chemist, but not by us.
No, I will not be looking at that poop or that email.
You can also sling some, ping us on Twitter, where we are at TWIScience, at Dr. Kiki, at Jackson Flying, at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in 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 Science.